So I work for the National Cancer Registration and Analysis Service. So I'm going to talk about a little bit about who we are and what we do, and then give you some data on the registration of GISTs and show you our headline figures over the recent years. But the main content of my talk will be trying to demystify some of the genetics and molecular biology and to explain why this is important in all cancers and how that relates to GISTs. So it's going to be doing some of the very basic biology and talking a little bit about molecular classification of GISTs. So my disclaimer is that I'm not an expert in GIST at all, and I'm not a medical doctor. I'm a genetics scientist, and that's my background. So the National Cancer Registration and Analysis Service, we were separate services around the country, and about 6 years ago we all came together to form a national service on one single system. So we collect data on every case of cancer that is in somebody who is diagnosed or treated in an NHS hospital, and we've got legal privileges which enable us to do this. So you can see here, this is the scale of what we're dealing with. We collect data from all the multidisciplinary teams in the hospitals, all the NHS healthcare providers, and you can see at the bottom all our different types of data feeds, so including the pathology, the molecular, the radiology. So we've got a complete record of everyone's diagnosis, pathology, treatment, and their outcome. So it's a massive, massive system. And this is for all types of cancer, not just GISTs. So I'll talk a little bit about what we register in terms of GISTs, because I know questions have been raised before. So we're constrained by what we can do under the law, and this is our little bit of the law, section 251 of the NHS Act 2006, for anyone who wants to look that up. It's a particularly exciting read. And it's all written in legalese, as you can imagine. So we can register all confirmed or suspected cancers diagnosed or treated in the NHS. And in determining whether something is malignant or not, we follow the guidelines from the Royal College of Pathologists. So we register all GISTs that are classed by RCPath as malignant or of uncertain behaviour. But we're not allowed to register GISTs or any tumours, in fact, if the pathologists' national body classes them as benign. So in practice, if we saw Philippe's table earlier of how GISTs are classified, so that would just be the top row of that table that we cannot register because the pathologists consider those to be benign. But everything else we know about, we register, so we've got a pretty accurate record nationally of what's going on. So that the benign ones are the tiny little ones, they're under 2 centimetres, and they have what we call a low mitotic index, and all that means is it's a measure of the rate of cell division, so they're small and they grow very slowly. So one of my analytical colleagues kindly pulled out this data for me. And this is age standardised incidence rates for GIST. This is just for England, and we've got the figures here for the years 2012 to 2016 inclusive, so over this 5-year period. So you can see that over 5 years that there have been fewer than 4,000 GISTs diagnosed. So it's fair to say that GIST is a very rare type of tumour. And last night, I pulled out the comparable figures from CR UK for bowel cancer, just any tumour type. So in 2015, there were nearly 42,000 cases of bowel cancer, and that's just for 1 year, whereas you can see for GIST over 5 years, we've got a much, much smaller figure. Clearly the GIST figures are for England, the bowel cancer figures are for the whole of the UK, but you get the picture anyway. So of those GISTs that we've got on the registry, we can look at them by age, and I don't think this will be a surprise to anybody. So that they really are particularly rare under the age of 35, but it happens, as we know, sadly. And the majority are diagnosed in people in their 60s and 70s. I don't think that will be a surprise to anyone. And then we can also break it down by where the GISTs were diagnosed and their behaviour. So the ones I've coloured in pink are the ones that are malignant, and we've broken those down into the different sites. So as you can see, the majority of those occur in the stomach or the small intestine. And then at the bottom in blue, we've got a large number, well, relatively large number, which are of uncertain malignant potential. And you can see that most of them do seem to be classified this way. So I think it's quite good to see these figures, and it just gives us an idea of what's happening at population level. And that's the beauty of the cancer registry. We're not just dealing with a subset from a research study, and research studies can be biased in terms of the patients they recruit. This is real population data, so this is what's actually going on, and that's the beauty of what we can do in the cancer registry and the power of this data. Okay, so I'm going to talk a little bit more now about DNA and cells and genes, and just go through some of the basic biology. So hopefully you know that we're all made of cells, and every living thing is made of cells, and we start off with an amoeba who has one cell, and then we have the nematode worm who's got exactly 959 cells. And I'm not joking, some scientists spend their whole careers working on these nematode worms, and they can trace the lineage of one of these cells exactly where it goes from. And here's a human who has about 10 to the 14 cells, and actually this particular lovely specimen of human did spend his career working on nematode worms. This is Sir John Sulston, but he did a lot more than this. He's sadly passed away a couple of years ago, but he was one of the pioneers with the Human Genome Project. He was a very vocal advocate of getting the genomic data out into the public domain, whereas other people and big corporations really wanted to privatize that data and keep it to themselves. So John Sulston is a real hero of a geneticist. So the genome, the DNA, is kept in the nucleus of each cell, and this is like the inner sanctum of the cell. It's kept there separate, so it doesn't get corrupted. And there's about 2 meters of DNA in each cell. And we've got 2 copies of the entire genome. And we inherit one from our mum and one from our dad. And then the protein synthesis, which I'll talk about later, that happens in the cytoplasm, the main body of the cell. So it's all kept quite separate. Here's the human genome. This is an even better read than Section 251 of the NHS Act 2006, a rather longer one as well. If you've got a spare 100 years or so and you find yourself in London at Euston Station, if you pop over the road to the Wellcome Trust, you can see this and you can read it. And this is 3 billion base pairs of DNA, and then we multiply that by 2 because we've got 2 copies each. And there's about 20,000 genes, and between any 2 of us human beings, we're about 99.8% sequence identical. So there's 0.2% differences between us, which works out at about, I think, oh gosh, I did the maths last night, and I think that comes to 6 million, but I can't remember because I was up a bit too late last night. So, as you know, we're all a little bit different. Some lovely diversity in the human species. And the reason for this is because we all have variations in our DNA. And here's a couple of, these are just normal variations. This is similar to the DNA sequencing that Philippe showed you. And you can see the highlighted bits, the blue traces and the green ones on top of each other. And that's because we've got one from mum, one from dad, and it's a little bit different at that position. And these are sometimes known as polymorphisms, which just means many forms. So we all have these, and that's what accounts for human diversity. So the other diversity, of course, that we see is all the different cell types we have in the human body. And when you think about this, each cell has the same genome in it. So how can we have all these different cell types? Well, the reason is it's all to do with something we call gene expression. And this is whether a gene is switched on. So there are some genes that are switched on in all types of cells. These are called housekeeping genes. These are your really important kind of bread and butter functions. And then there are some more specialist genes. So, for example, the liver enzymes, they only need to be switched on in liver cells. You don't need a liver enzyme to be active in one of your eye cells, for example. So that's how cells are very specialized for their particular functions. It all depends on which genes are switched on in those cells. So what we need to remember here is that each cell contains 2 copies of the genome, and the behavior of a cell depends firstly on the DNA sequence of its genes, and secondly on which genes are being expressed or switched on within the cell. And these are quite important points. So what's a gene? Well, it's basically a recipe. That's all it is. So here we've got the nucleus of the cell, and the whole genome is like the whole recipe book, and I've got Mary Berry's lovely complete cookbook here. So that's the whole genome, and one gene is a single recipe in that book. Now, the nucleus, it's a little bit like, I think most of us are old enough to remember reference libraries. Of course, everything's online now, but back in the day when you had your reference library where you could go in and you could consult the stuff in there, but you couldn't take anything out. This is how the nucleus works, really. It is an inner sanctum. The DNA is kept nicely in there. So this process called transcription makes a copy of the gene or the recipe. This is like when you copy something out of your granny's recipe book by hand, like they used to do back in the day. And that's called messenger RNA. So that's a little copy of the gene that can then go out into the cytoplasm. And then the protein gets made. So the gene is the recipe, and the protein is the actual dish. And the amino acids are the components that get put together in a certain order to make up the protein. And the gene tells you how to do that. So let's think now about a recipe, and perhaps we're making a nice ham quiche here, and this is what our recipe tells us to do. And in the genetic language, well, there are 4 letters, but each word consists of 3 letters. So it's all 3-letter words in the genetic alphabet. So let's introduce some spelling mistakes into our recipe now. So the first one, if we add 1 egg, add the ham, well, that doesn't make any difference at all. That's completely harmless. That's doing nothing. And in genetics parlance, we would call that a synonymous variant, but you don't need to worry about that. And then the next one down, we get add 1 egg and the jam. Well, that's an unusual variation on a ham quiche, but actually it's perfectly edible. It might be perfectly nice. It's not going to do you any harm at all. That recipe will work. So that is what we consider to be a normal variant, and those are one of the things that just accounts for human diversity. The next one down, we're coming into a bit more trouble, because if we put a hat in our recipe, that's probably not going to work so well. And we would call that a missense mutation. We would call that pathogenic or disease-causing. That's going to cause you problems. And then the next one down, we might lose a little bit of the recipe. That's an in-frame deletion. And then the next one down, we've lost just 1 letter from the recipe. And this shifts the whole reading frame of the recipe over by 1. So you end up with this complete and utter nonsense. It becomes unreadable. And that's a pretty bad mutation to get in one of your genes. You can see that completely messes with the function of the gene. And then you can get duplications, you can get insertions, And then at the end, you can get this where you get a premature stop instruction. So at the end of every gene, there's a 3-letter word that says stop. And if you get that word earlier in the gene, then the protein gets truncated. So all of these, so the 2 at the top are normal variants, and the rest of them are pathogenic or disease-causing variants. So let's go back to school biology now and think about our old friend, the amoeba. So remember, here's the amoeba. He's got his DNA within his nucleus. And if we mutate the DNA in that amoeba, if it's one of these really horrible mutations, the amoeba might die and the mutation will disappear with it. If it's one of the neutral mutations, the amoeba won't even notice he's got that mutation and it'll carry on perfectly happily. There'll be no effect whatsoever. If it's an advantageous mutation, then the amoeba can pass that mutation on to all its amoeba babies, and it grows faster, it has more babies, and over time that mutation becomes more prevalent within our population of amoebas. And this is Charles Darwin's theory of evolution. So that's what's going on in the amoeba. Now let's, instead of an amoeba, this is now a human body cell. And remember, in the human body cell, each cell has its own copy of the DNA. So let's think about the same sort of things. If we get, for the cell, a disadvantageous mutation, the cell dies and the mutation disappears with it. And that's fine. We're human beings. We can afford to lose a few cells here and there. That's no problem. It's a problem for an amoeba, which is only single-celled. It's not a problem for us humans. If we get a neutral mutation, there's no effect on the cell. So, for example, if I get a mutation in a liver enzyme gene in one of my eye cells, well, the eye cell doesn't care, does it? Because it's not using that liver enzyme gene anyway. So it doesn't matter. So all our cells have some of these mutations in that are doing us no harm. If the cell gets what it thinks is an advantageous mutation, that cell will grow and divide faster. And the mutation is passed on to all the daughter cells. And this is the first step in the development of cancer. So this might be in a gene that is promoting growth of the cell. So this isn't actually a tumour yet, but as it acquires more mutations, it may become cancerous. So tumours evolve over time. And actually, I think this is a really nice diagram. This is Charles Darwin's original notebook from 1837. And then in 2012, they did some whole genome sequencing. This was actually on a kidney cancer. And they could track all the mutations as they happened in the kidney tumour. And I think the similarity between what Darwin predicted for evolution of the species and what's actually going on in this tumour, I think the similarity is really striking. So, yeah, if Darwin had had a DNA sequencer, he'd have found something similar. So he was quite a genius of a man. So how do we treat cancer? Well, the old way was basically surgery, radiotherapy or chemotherapy, cut it out, burn it out, or poison it out. And they're primitive. We've got a surgeon talking to us later, so he'll tell you about his primitive work. But they are very successful approaches. The trouble with chemotherapy, radiotherapy, they do kill normal cells as well as cancer cells. And this is why chemotherapy, the side effects are so brutal. They kill anything, any cell that's dividing, which includes the cells lining your gastrointestinal tract, which is why chemo causes such awful nausea and vomiting. Your hair follicle cells divide very fast, so you lose your hair. Your immune system cells naturally divide very fast, so chemo can make you immunosuppressed. So they're not nice treatments. So really the way forward with cancer treatment, what we're moving to now is personalised medicine. And this takes into account the DNA sequence of the human and the DNA mutations in the tumour within that human. So you can extract DNA from the patient and from the tumour. You can compare the DNA sequences. You can profile the mutations. And then you can prescribe a drug or a combination of drugs that are targeting the specific mutations in that tumour. So now we start to think about mutations in GISTs. Okay, and when we're thinking about mutations, we need to think about two different classes of mutation. So the first mutation is called a germline mutation. That's one that is present in every cell of your body. And the reason for this is usually because you've inherited that mutation from your mum or your dad. And germline mutations cause rare diseases because they're in every cell of your body, and they can cause rare inherited cancers, but most cancers are not inherited. And you can detect that mutation in somebody's blood, and if they have a tumour, the mutation's obviously also there in the tumour DNA. The ones we're more interested in cancer are what we call somatic mutations, and those are the mutations that occur just in individual cells, so they're only found in the tumour. And you need multiple somatic mutations in the same cell, and that's the underlying cause of all cancers, whether it's a breast cancer, a bowel cancer, a GIST, a lung cancer, anything. They are all full of somatic mutations. And you can detect these mutations in the tumour DNA, but not in the blood, unless you're dealing with a leukemia, in which case it gets complicated. And that can cause some technical issues. And it's an important distinction. So if we take the KIT gene, for example, if you have a germline mutation in your KIT gene, so this is a mutation in every cell of your body, and this mutation, this particular mutation in KIT actually makes KIT lose its function. And this is the cause of piebaldism. Did anyone know that? Did anyone know that KIT's related to that? So here's a nice piebald horse, and here we've got a mouse and a baby with a similar pattern. And this is because the KIT gene is really important in embryonic development, and it gets some of the cells to the right place when they move about during formation of the embryo, including some of the pigment cells that produce melanin. So that's why the pigmentation pattern is not right if the KIT gene is not working properly. So that's an underactive KIT gene in every cell of the body. On the other hand, if we're thinking now about a somatic mutation in the KIT gene, and it's also a different type of mutation, so it's a different type of spelling mistake in the gene, and this doesn't make the KIT gene underactive, this makes it overactive. So let's think about what the KIT gene does, and PDGFRA works in a very similar way. So the KIT gene, it's what we call a growth factor receptor. And when this stem cell factor, this growth factor, binds to KIT, then KIT sends a signal to the nucleus of the cell, and this tells the cell nucleus to switch on growth genes, and that causes the cells to divide, and it causes the cells to move around and migrate a bit. And all cells need a few growth factors to keep them healthy. This is cells just talking to each other, communicating, checking in with each other. And extra growth factors are needed at certain times, like during embryonic development or during wound healing, for example, when the tissue needs to be repaired. But when the growth factor is no longer needed, that's gone away. And you can see here that KIT is switched off, so it's not causing those growth genes to be switched on. So in a situation of cancer, where the KIT has a mutation in it that makes it overactive, what this means is that even in the absence of the growth factor, KIT is permanently signaling to the nucleus, and it's permanently giving it this message to switch on the growth genes. And that's why the tumour develops, because these cells are growing and dividing out of control. So, of course, what we can do now that we understand the biology of these tumours is we can treat them with a class of drug called a tyrosine kinase inhibitor, and here's Gleevec or imatinib. And actually, Gleevec was developed to treat chronic myeloid leukemia, which is a different sort of mutation in a different gene. But we know that the biology of those tumours is very similar, and they're both KIT and the one in chronic myeloid leukemia, they're both called tyrosine kinases. They're this class of molecule, so the same sort of drug can work on both. So this is a nice example of repurposing a drug from one type of cancer to another, just by understanding the molecular biology of that tumour. So that's why we do molecular biology. That's why it's so important. So the work that I'm doing at the moment is collecting all the molecular testing data from the NHS labs around the country, which is quite a big challenge. And one of the major challenges we've got is that all the information systems in the labs, all the lab databases, computer systems, most of them were kind of built in the 1980s, as I'm sure Philippe can attest to, really before any of this molecular work was done. So they are not well set up at all to capture this information. And every lab around the country is using a different system. So we get data in from each lab, and this is highly technical molecular biology data coming in from each lab in a really different format. So we've got the technical challenge of trying to get that into one standardized format. And we've also got the challenge that our registration staff, who are their admin grade staff, they're absolutely brilliant, but I've had to train them up in postgraduate level molecular biology so that they can interpret this genetic data and record it accurately in the cancer registry. And we have cherry-picked some stuff to do that, and I'm so proud of them. They're doing absolutely brilliantly. I'm completely blown away with their capability in this area. And then we can develop code in this SQL, structured query language, and that's what my analytical colleagues do. And then we can look at the data and analyse it. So this is where we're up to nationwide with collecting molecular biology data from NHS labs. And this obviously covers all types of tumours, all genes. So at the moment we're registering data from the calendar year 2016. We've got 7 labs all registered. We have got data from a few more labs, but we're still processing that. And I think you can see the amount of work that our registration staff have put into this. Yeah, they've registered nearly 60,000 genetic test results. And just to show the diversity of the sort of genetic testing that goes on in NHS labs, we've got 1,099 combinations of gene and tumour site. So it's incredibly complex work, it's pretty painstaking, and it's quite novel work, and I don't think there's anywhere else in the world that's doing this to quite the degree of granularity that we're doing it. So this is hot off the press information about the mutation status in GISTs. So as you can see, there's only a fairly small number. We've only got 255 in there at the moment, but this is a new work stream. Remember, this is just for the year 2016, and we haven't yet registered all the data from all the labs. So, but I think we're getting there, and I think this shows proof of principle. So you can see that about 3/4 of the GISTs do have a KIT mutation. And then you can see the ones with the PDGFRA mutation, and then all the others. And the yellow ones are where no mutation has been identified. So those are the ones that we would call wild type. So as we get more of this molecular biology data, we can compare it to patient treatments to make sure that people are getting the right treatment. We can compare it to outcomes, and we can really start to drill down into how the biology of the tumour is affecting outcome and survival and so on. And as next generation sequencing comes on board, we can get more and more data. What we're doing at the moment, all this reflects targeted testing, where the labs have cherry-picked genes and they've said, okay, we're just going to test KIT and PDGFRA, we're not going to test any other genes. But with the next generation sequencing, a lot more genes will be looked at, and we'll start bringing in this data as well. So it's really early days, but this has the potential to be a very, very powerful data resource. And as I say, it is population level as well, so it's an unbiased sample. So this is really early work, but I'm excited by it. So thank you for listening, and I'm happy to take questions either now or in the panel.
Yeah, thank you very much, because it was very instructive to go through the process of the genetic mutation, and very good education for me. Thank you. I saw that in your slides you mentioned that you don't collect data from London. Is there a reason why there's no national database of genetic mutations? And the second question is around malignant GIST. So you record the genetic mutations, but do you follow up also all the treatment for patients so that you can see which treatments are given to certain type of mutation or GIST mutation? Or
yes, so I think the question of, so I guess there's three questions. The why isn't there a national database of genetic mutations? Well, that's exactly what we're trying to create, and it's technically a massive challenge. The second question about the gap in data from London, I think part of that is just because the testing landscape is so complicated in the London hospitals. It's been harder to get a grip on that, and perhaps we've had a bit less engagement from some of the labs in London. We are working to address that, but I think by the nature of things, you go for the easiest targets first, and we've asked everyone, and then we've worked more with the labs who are more keen to give us their data, but we'll get to London. It's a gap that we know about and we are addressing. The other question was about treatments. And
so I understand the database is recording the information about the gene mutation, but is that database as well including information about the treatments that the patients are receiving and the prognostic of survival?
Yes, so it is recording all that. It comes in through different data sources. So I get the data directly from the molecular biology labs. Oh, and it's worth saying as well that we're not just recording that KIT is mutated, we are recording the exact mutation in the KIT genes. So although I haven't shown you the exon 11 mutations and the exon 9 mutations, that information is all in there, and we do have the capacity to be able to pull that out. So that's the molecular side. The treatment side, that comes in through what we call the SACT database, which is the Systemic Anti-Cancer Therapy database. And this is a national database that records all uses of chemotherapy and all use of targeted therapies, such as imatinib and so on. So that comes in from one data feed. And then we get the outcome and survival data through the Office of National Statistics and the registration of deaths and so on. So basically what we're doing is we get different data feeds from all over the NHS, from all over the labs, from national databases, from local hospitals. Anywhere there's data, we pull it in and we link it all up at patient level and at tumour level. So yes, we can correlate everything. So hopefully that answers your question.
You mentioned the legislation, and it only allows you to record information on malignant cancers, not benign.
Yeah.
Would it be useful to have the information on benign, and could the patients provide you with the OK under the GDPR, the authority to record that information?
I think if the patients wanted us to have this information, and I am thinking on my feet here because I'm an expert in genetics, not information governance, I don't think there would be any ethical or legal problem with that, but the problem I can see is more a host of practical problems in that our systems are set up to collect all the data on known malignancies. And of course, the other thing is if it were an opt-in thing, so people with benign conditions could say, oh, I want to go on the registry as well, we would not be getting that at population level. You'd just be getting a few bits and pieces there, so the data wouldn't be very meaningful. So I think probably ethically not a problem, practically a massive problem, I think.
Right. Oh, is it possible we could keep the next questions to the Ask the Experts? Could you write them down for later, because we're kind of running over time?
Yeah, sorry, I will be here on the panel later.
Okay, thank you very much, Fiona. Well, hope...